Okay, I'm Ken Leonhardt. My title is Breeding Leucospermum for a Warming Planet. I'm with the University of Hawaii. Oops.
Let me switch it to the other side since you're going to be turning it kind of on.
Okay,
That'll help.
thank you. Okay, breeding for heat tolerance. Climatologists tell us that the planet's getting warmer, so let's get ready for that. Governments are promoting the use of low water requiring plants, and we all know that Proteaceae in general are drink responsibly. California, Arizona, Texas, Florida, largest markets for Southern Hemisphere genetics according to our friend Kruger, Peter Kruger, who made this presentation at a new crops conference in Perth in 2015. And so these are motivations. At the University of Hawaii, we've been breeding tropical proteas, breeding for tropical proteas since 2001. And since my research, whether it's protea or landscape trees, is always in the interest of Hawaii growers. So Hawaii growers might find markets in several southern states for University of Hawaii tropical protea varieties. I started in 2001, I started doing tropical protea varieties as a result of recognizing that there was more agricultural land available and less expensive than the higher elevation lands where protea is traditionally grown, because sugar and pineapple have gone out of business. So I thought, well, it would be great to be able to grow proteas at those elevations. So while I was still doing breeding work at the experiment station on Maui at 3,000 feet, I was looking for species or hybrids that would perform well at low elevation. So I started doing some potted plant trials at Maui Community College at an elevation of 50 feet, and another tourist company called Hawaii Tropical Plantation at a couple hundred feet. And there were some encouraging results. Then we lost funding for the work that we were doing at Maui, and which was okay for this kind of a project, because you can't screen for warm temperature tolerance at 3,000 feet. So I brought the project over to Oahu, where I am, where I have my laboratory and home on Oahu, and I set up two trials. One at the Waimanalo Research Center, research station on windward Oahu at about 50 feet, and then one in Kunia in central Oahu at about 200 feet, 220, 30 feet. This is that particular plot, and this is, if I can point at this, this is a portion, oops, whoa, wrong button, whoa, wrong button. There we go. This is actually Pearl Harbor. Most of it is out of view on the right. And this, where is it? Somewhere over here is Diamond Head. Do you see it? I saw it earlier, somewhere over there, Diamond Head Crater. Anyway, so this is the plot that originally started. I had a graduate student from Costa Rica, Thomas Littleton. He was interested in this project, so this was his master's research. This is the Kunia station after about a year and a few months. And this is where we started making observations. A lot of our, this is hybrid 29. This was an outstanding hybrid for breeding. It was one of the real early releases by the University of Hawaii. And here it is grown at the experiment station on Maui, and then over here at one of our low elevation sites. And this is what most of the hybrids did at low elevation. They just couldn't get off the ground. They just sprawled around, and there were two independent responses. They had erect growth habit, or they didn't. They sprawled like this. They flowered, or they didn't, and we had all combinations of that. Of course, we were selecting for plants that had an upright growth architecture and flowered. And here's another one. This is hybrid 79. It did the very same thing. Someone described this plant as appearing to be an intoxicated serpent. So how do you select for warm temperature tolerance? We were using 13 different species in our breeding program. This one I think has 9 species in it. This was be sure I'm using the right button here. This was peppermint. This proved to be a very a good selection for warm temperature tolerance. It had upright growth habit, and it flowered quite freely, very similar to the control condition at Maui. And it had, so how do you sort this out? You know, to find out where you get into warm temperature tolerance. Well, and this is just one example of many of our hybrids that had anywhere from 2 or 3 or 4 species up to 11 or 12 species. So I thought, this is what's called genome breeding, not genomic breeding in the sense of molecular breeding, but genome breeding in the sense of which genomes are more closely related. And taxonomists will look at a genus and divide it into subsections called sections, taxonomic sections. Up here, taxonomic section. And the 48 species of Leucospermum are divided into 9 taxonomic sections. And all the species within any given section are more closely related to each other than they are to any other species. So I've highlighted 3 sections here, Conocarpodendron, Brevifilamentum, and Cardinostylus, because those are the sections that accommodate pretty much all the species used in breeding Leucospermum hybrids. And I've verified that from looking at all the registered hybrids in the what is it called? The hybrid registry. So let's see if we can sort this out. I use a program, a statistical analysis program called Principal Component Analysis, and all of these 3-digit numbers represent a hybrid. And I inputted all the genealogical details from those charts, like the last frame that I showed you, into this program, and said, show me which taxonomic section is most closely related to the ability to flower at low elevations. So this is the axis for flowering at low elevations. This is the axis for the section Cardinostylus. This is the axis for the section Brevifilamentum. This is the axis for the section Conocarpodendron. So it's pretty clear that the taxonomic section Cardinostylus is where the genes for warm temperature tolerance are coming from. Okay, good to know. Let's go back and look at that thing. Here we go. Cardinostylus. We're looking at these 6 species. Now, the ones highlighted are the ones that were available to me at the Maui station where the breeding work was done. Formosum, Reflexum, and Grandiflorum. That's what we had to work with. And so that's what we did. Let's just have a look. Section Cardinostylus, the ones that we had, the 3 out of the 6, are Formosum, Reflexum, and Grandiflorum. This is what they look like individually. I think as species, I think they don't have a lot to recommend themselves by. I know Reflexum is used as a cut flower. Frankly, I'm uncertain why. I don't think I think it has more undesirable characteristics than it does desirable characteristics as a cut flower in itself. But it's got warm temperature tolerance. It's got a really long stem. And everything else, the other 5 species in Cardinostylus section have short stems, thick, big diameter stems, heavy leaves, just a lot of the undesirable characteristics. But they have warm temperature tolerance. That's what makes it all fun, because you got to make the hybrids and sort through it. And some other noteworthy things. Formosum has tolerance to PCs. Whenever you see it on my illustrations here, it's Phytophthora cinnamomi. And in Hawaii, and I think possibly worldwide, it's the single biggest problem for root disease. Cylindrocladium is a problem also, but I think Phytophthora is a bigger problem. Reflexum has some tolerance to PC. Grandiflorum, we don't know, but we know it comes from hot, dry slopes of the Parle Mountains. Why do you think Formosum has Phytophthora tolerance? Of the 48 species, it's the only one that is found growing in a bog habitat, okay? Wet feet. We know that Protea don't like wet feet, but here's a species that grows in a bog habitat. Good place to look for Phytophthora tolerance. Okay, let's look at Formosum hybrids. The first one to look at, I should have reversed these, but the first one to look at would be a spider. This was developed, this was made, I don't know if Gert Britz made it or Gail Littlejohn made it, but one of the South African breeders made this, and it's used widely in South Africa as a rootstock because of its tolerance to Phytophthora. We had it, so we put it in the breeding program to see if the Phytophthora tolerance was heritable or not. Here's one of our hybrids, 561. Formosum times linear glabrum turned out to be a really good one. Let's look at some of the Formosum hybrids we made. 262, I already showed that to you on the genealogical chart. It only has 12.5%. So one grandparent was Formosum, but I don't remember if we tested it for PC tolerance, but it does have warm temperature tolerance. It produced a really nice heavy textured flower on a nice long stem. It's got good colors. It's something that we thought was really good. This is a hybrid 100. Also, this is 25% reflexum, and it was a real good one for us. Here's another Formosum hybrid. This one also only has 12.5% Formosum, but it's got good warm temperature tolerance and good upright growth habit. It also has 25% Saxosum. I started, when I first was looking for a species that had warm temperature tolerance, I identified Saxosum as a probable species for warm temperature tolerance. Why would that be? It's the most equatorial species. You look at where are these things grown. I read Tony Rabella's work, and where are these things grown? This one is grown, all the others are grown in, native habitat is in South Africa. This one is in Zimbabwe, okay? Closer to the equator, more equatorial. Obviously, wouldn't that be obvious? Should be more warm temperature tolerant. Well, I used it for a couple years, and I'm thinking, where's the warm temperature tolerance? Because it wasn't, it didn't convey, it in itself was not warm temperature tolerant, and it didn't convey any warm temperature tolerance. And I thought, I don't understand why this isn't the case. Well, if I had done a better literature review, can I point this right there, my grad student, Ashley. It's the importance of just putting out the importance of doing your literature review, because had I done a better literature review, I would have realized or would have learned that Saxosum, even though it's the most equatorial of all the species, it's from high up in the mountains and it's not warm there. So there's no reason to expect warm temperature tolerance. Okay, let's move on. Reflexum, long straight stems, small narrow leaves, warm temperature tolerance. That's the, by the end of this presentation, I'll figure this thing out. So the importance, the two important things here, warm temperature tolerance and these are all important, small leaves and long stems, because the other species in Cardina stylis don't have those characteristics, and that's what the growers want. So look at Reflexum hybrids. This is one that's become quite popular. People like Hybrid 74, Grande de la Cruz. This is June. These are both 50%. These are both hybrids with Reflexum as one parent. They're really, really excellent hybrids. Another couple more with 50% Reflexum. Oh, here I thought, okay, Ken, let's do something really cool, and let's cross lineary and Reflexum, both of them out of the Cardina stylis section, both of them with really long stems, both of them with crappy flowers. And I expected to get a hybrid with crappy flowers, but a plant that I could use in breeding, and it would segregate from that particular parent because it's a hybrid. And and so we've used it that way. Here's another one, Hybrid 145, reflexum times a cross of Conocarpodendron and cuneiforme, and it has really good warm temperature tolerance. It doesn't have really long stems, but it's a really nice plant. It's a heavy textured flower, got PC tolerance. That says resistance. I really hesitate to use the term resistance. I'd rather use the term tolerance, because it doesn't mean that you can't, doesn't mean that it won't come down with it. It just, okay, let's move. Reflexum hybrids, here's another one that's half reflexum, 20. Oh, what's interesting here is that pretty much all the populations that were half reflexum, the lower styles would reflex like this, and you're familiar with that. Anything less than 50%, like if it's 25%, they don't do that, or they don't do it as much. These are probably a little more difficult to pack, or you have to pack them in a little more closed bud, or harvest them in a little more closed bud, unless you're going to just put them in a bucket and run to the local florist. And they're usually easy to propagate, and they often have PC tolerance. These are 25% reflexum, 570 and 162. My wife and I are going to go and visit Georgette after this conference. She's our middle daughter, lives in Washington. She's got 2 babies. Brandy Delacruz. Oh, both of these are Brandy crosses, Hybrid 74. Really successful crosses for successful hybrids for low temperature tolerance. By the way, you'll hear more about the tropical Proteas from Russell Galanti, our county agent for Big Island floriculture crops. He's going to talk later in this meeting, and so is Jamie Barton, who is our first commercial grower, growing, so the grower's perspective on this. Okay, let's have a look at grandiflorum. I like grandiflorum. It's just, I personally like this little swirly styles and the gray, bluish kind of tips of the styles. Here's a hybrid of, and sometimes it's heritable. I guess it depends on what you cross it with. You see it in some populations, hybrid populations, and others you don't. 2 more grandiflorum hybrids. They tend to be pretty large flowers. They tend to not have real large stems. They tend to have larger leaves, which I really don't like. I like small leaves. I like leaves with a lot of space in between the leaves. Why? It's a means of allowing the plant, it's a means of letting the breeze flow through the plant, dry off the plant quickly, so that you don't get infection by foliar fungal organisms. The growers don't want to spray it, and nobody wants a grower to spray it. And so if you can use a method like this of just developing the kind of architecture of a plant that will dry off quickly, you're less likely to have foliar fungal problems. 2 or 3 hours of free moisture on the foliage and you can get germination of fungal spores. Less than that, you're less likely to have fungal spore germination. Crosses, this is fun. This is really a fun part. When you bring in Cardena stylus species from both sides of the cross, from both your paternal and maternal parent, and that's what I've done in this case. We got Formosum on the maternal parent and Reflexum on the paternal parent. This is Hybrid 51, one of the first ones. I like it. I don't know. I don't know what else to say about it. I like it, but it's got some deficiencies. 152 is another one that the product of crossing with using parents with Cardena stylus species from both sides, maternal and paternal. Hybrid Lin, got Formosum and Totem, Formosum from one parent and Reflexum from the other parent. Same with this one, Hybrid 207. This one is really good. I mean, I personally like this one a lot. It's got a nice color, really firm, heavy textured floral segments, and acceptable foliage length. I haven't said much about lineary because it's not from the Cardena stylus, but it's a species that I've used, or hybrids of it, when crossing to the Cardena stylus hybrids, because it has long, really long narrow and strong stems with really, really small foliage. Foliage, the species itself, or the selection of the species we use, starlight, has foliage that is very similar to a pine needle. You know, it's like a needle-like foliage. Okay. What slide number am I on? 24 of 40. Okay, we need to shift gears. Hybrid, what are we doing? Oh, this is grandiflorum with reflexum. Okay. Bolero and 29. This is also reflexum and grandiflorum. Well, yeah, the species grandiflorum as the paternal parent with the hybrid. Oh, here's 29 again. Remember Rachel? She was one of the best breeders. I say she. It's the only plant, the only hybrid that we used, and we use it extensively, where we get a different population of plants when used as a female parent from when we used it as a male parent. You normally don't, that's an unusual situation. It means there's something irregular going on in meiosis. But it was fun. All of the selections that we introduced using hybrid 29 Rachel as a parent were when we used it as a female. And all the hybrids that we raised from it when Rachel was used as a male went to the compost pile. So these are 29 reflection times 633 at the, at 30 meter at the Waimanalo Experiment Station and at Cunha, the two low elevation sites on Oahu, and they performed really, really well. Okay, grandiflorum reflexum again. Okay, and this is just to illustrate how large the flowers are. This concludes the progress report part of my presentation. So what do we do about the future? There's so much that hasn't been done, and I really wish I had started Protea breeding when I was a young faculty member. I didn't start until I was in my 50s or late 40s, I don't know. But there's not, okay, these are opportunities. The International Protea Register, 11th edition, only 150 hybrids. And then 6 years later, the 12th edition, only 7 more hybrids. All of them introduced, registered by Future Finboss. Their last registration of a Leucospermum hybrid was 2018. So there's not much going on in terms of variety development. Or I should say, if there is, they're not being registered. I'm taking this data from the registration. There's 48 species. Only 16 have been used in breeding, and only 9 of them have been used extensively. 15 species in 2 different sections are fragrant. Why don't we have, why are there no white, blue, lavender, or purple hybrids? Why are there no polyploid species or hybrids? Why are there no intergeneric hybrids? These are all things that I believe are possible, and I'm about to show you why. Okay, here's those taxonomic sections again. Okay, I'm not going to read them off. It's too. Here they are. Here's the 16 species that have been used in hybrids that have become registered hybrids with the International Protea Registry. It's a little, I don't want to say misleading. There's 16 have been used, but Saxosum only twice. We used it once. Uberescens only twice. Praecox once. That's our hybrid. This one once, Formosum once, that's the South African one. When I do get around to registering some of our stuff, our warm temperature stuff, that there'll be more Formosum hybrids. So only 9 out of 48 have been really extensively used. This column has species that have never shown up in the registry, so they've never been used. Or like I say, if they have been, nobody knows about it except the breeder. Okay, what's highlighted here? The yellow ones are fragrant. Oh, I made a mistake here. Sorry, I didn't catch that. Tony Robello, that's an L, not a K. I'll correct that. The yellow ones are fragrant. Every species, there are 15 species in the section Leucospermum and the section Diastyloides. This section, let me see, I want to be sure. It's the genus Diastella. Okay. These are all fragrant, okay? And if you notice, only one has been used to make a registered hybrid. And there's 14 others that don't show up in the hybrid registration, but they're fragrant. So why don't we have fragrant Leucospermum? Here's the one. Okay, I could not find, I contacted Tony Rebello and I contacted other people. I contacted Kirsten Bosch Botanic Garden, which it was known to be at, and nobody seems to know where this plant is. Thompson's Gift is a registered name. It's across the Concarpodendron times hypophylocarpodendron. And it's fragrant. What does that tell you? It tells you that the genes for fragrance are heritable. Okay, you can make a cross with one of these species, and the gene, at least for this cross, I don't want to make a broad statement about it. Here's the section diastyloides. Everything was sweetly scented. Everything has fragrance. Okay, there's the opportunity to incorporate fragrance into commercial hybrids. Well, why aren't we doing that? Well, I know some of you are thinking, Ken, you're just not thinking clearly. These things are so small, they can't make any significant contribution to commercial varieties. Quit pretending you're... And I said, well, you know what? I'm holding 40 tomatoes in my hand. Okay, this is Solanum pimpernellifolium. And my friend, the late Dr. Jim Gilbert, was a world-famous tomato breeder. Nobody had hybrid tomatoes with nematode resistance, but he found this plant in the Andes, and it's got nematode resistance, and over multiple generations, back crosses, sibling crosses, on and on, he made a whole bunch of varieties, new varieties, table varieties, that have nematode resistance, and it came from this little species. So no more discussion about how small those plants are. What about intergeneric crosses? There's a known hybrid of these two plants. This is Diastella familioides and Leucospermum oleifolium, both South African plants. And it's documented that there was a hybrid, a natural occurring hybrid. Tony Rebello told me that it did exist. It was propagated. It was sent to Kirsten Bosch. It was lost in a native habitat, and also it was not propagated and later died at Kirsten Bosch. So it doesn't exist. But the point is, you can make this cross of Diastella with Leucospermum. Yeah, Interesting, yeah? Well, why would you do that? Well, how about for pink, for lavender, for blue, for white? Those colors exist in Diastella. They don't exist in our commercial hybrids. What about polyploidy? All the important, I shouldn't say all, a lot of important crops. Peanut, banana, white potato, sweet potato, strawberry, coffee, sugar cane, cotton, they all have polyploidy. What does polyploidy do? It gives you, bulks you up, okay? Why? Well, every cell has twice the amount of DNA, if it's a tetraploid, twice the amount of DNA in the cell. What does that mean? That means tissues that are composed of cells are twice as large. What does that mean? That means organs are twice as large. What does that mean? That means that your whole plant, the fruit, everything, gets bigger with polyploidy. Polyploidy, we all know. What are the four major food groups that sustain human life? Sugar, caffeine, alcohol, chocolate. We all know that, okay? There's polyploidy in all those plant groups, all of them. So why aren't we doing that with, here we go. This is a triploid, cymbidium, and a converted, same clone, converted hexaploid. My student from, excuse me, just a second here. Oh,
It's over.
she says, it's over. That's management. I refer to her as management. Yeah, you're right, it is.